15 years of AIDS. The continuous failure in the prevention and treatment of AIDS is rooted in a misinterpretation of an inflammatory autoimmune process as a lethal, viral venereal disease. By A. Hassig, H. Creamer, S. Lanka, W. X. Liang, K. Stamfley, May 1998. Summary: the question of the specificity of the anti-HIV antibody test has to be re-evaluated, as it was shown that the viral enrichment obtained from co-cultivations of patients' lymphocytes with fetal cord blood by Barsanussi et al. And leukemia cells by Gallo et al. Exclusively consisted of proteins of the cell types used in the cell culture. This precludes a clear separation of presumed retroviral and cellular proteins or extracellular matrix proteins. In this context, it was shown that the anti-HIV antibody test detects autoimmune antibodies directed against cytoskeletal proteins, e.g. the liver cells. Strongly augmented anti-actin autoantibodies is considered close to pathognomonic for chronically active hepatitis. The original assumption that reverse transcription from RNA to DNA is evidence for the existence of retroviruses was wrong. In fact, reverse transcription is a vital mechanism for the maintenance of the genome. The decrease in numbers of circulating CD4 lymphocytes can be explained by a stress-induced hypercortisolism. Up to date, direct HIV-mediated destruction of CD4 lymphocytes could not be proved. The same is true for measuring of the viral load. Shortcomings of the applied method to quantify the viral load do not permit definitive conclusions. Possibly, it may be taken as an expression of a stress-induced weakening of the cellular immune reactions, in the course of which the nucleoside fragments resulting from the current cell turnover are inadequately eliminated. Furthermore, the treatment of patients with nucleoside analogues has a toxic effect on both the genome of the cell nucleus and the mitochondria. The latter, therefore, may produce insufficient amounts of ATP, causing organ failure and, eventually, death. The synthetic protease inhibitors used these days are associated with serious side effects. Therefore, it seems worthwhile in these patients to bring back the catabolic situation due to whole body inflammation to homeostasis by administering anabolic phytopolyphenolic compounds. AIDS is the abbreviation for acquired immunodeficiency syndrome. AIDS, as a term for an illness, originated in the search by the American Centers of Disease Control for sick homosexual men, also suffering from Kaposi's sarcoma, Kansas, and/or pneumocystis carinii pneumonia, PCP. In 1983, Barsanussi et al. Reported on a T lymphotropic retrovirus which they allegedly isolated from an enlarged lymph node of a homosexual patient. 1. In 1984, Gallo et al. Reported the alleged isolation of an identical retrovirus from CD4 lymph cells from homosexual patients, clinically diagnosed as suffering from AIDS. 2. Barsanussi et al. Co-cultivated these patients' lymph cells in question with fetal cord blood, and Gallo et al. Co-cultivated theirs with leukemia cells. Initially, these laboratory methods must raise doubts as to whether the isolation of a new human retrovirus is evident just by these data. Gallo et al. Stated that their allegedly isolated retrovirus caused the destruction of CD4 lymphocytes in those patients, whose heterogenic illness was taken as an after effect of the CD4 cell destruction, and so subsumed as AIDS. Besides, Gallo et al. Announced that in due time a vaccine would be available for the formation of antibodies against the discovered virus. 2. Today, 15 years later, the question still remains open, whether high retroviruses actually do exist, or whether the postulated retroviral HIV antigens as well as the postulated HIV reverse transcription are a matter of human protein molecules derived from cells in co-cultured cell cultures used by both Barsanussi et al. And Gallo et al. The most extensive investigations in this regard are owed to Eleni Papadopoulos and her group in Perth, Australia. In 1993 they published a review concluding that there is no evidence for the existence of high viruses. 3. In 1994 Lanka demonstrated that all retroviruses, including HIV, are biologically inexistent, and their phenomenology is based on laboratory artifacts. 4-6. Eleni Papadopoulos confirmed Lanka's interpretation in a recent comprehensive review. 7. These fundamental counter-statements to the current HIV/AIDS theory have been strongly supported in the last few years. Upon investigations in order to develop a vaccine against HIV, it became apparent that the enrichment of presumed HIV-1 preparations considered as pure consist of proteins of the cell types used in the cell cultures, which resist a clean separation into presumed retroviral and cellular proteins, i.e. extracellular matrix proteins. Above all, these cell proteins also occur in the inside of extracellular particles, which have been misread as it seems as so-called high variants by the retrovirologists, 8 to 10. These findings were to be expected as Gallo et al., when developing the AIDS test, did not investigate the presence of cell's own proteins in the protein mixture released during the co-cultivation of patients' lymphocytes and leukemia cells. Upon developing the ELISA and Western blot tests, it should have been imperative to consider proteins released from stimulated leukemia cells, not being mixed with patients' lymphocytes, and to differentiate these from the ones released only after addition of patients' lymphocytes. In view of this, it seems to be mandatory to reevaluate the question of the specificity of the anti-HIV antibody test. What is the laboratory finding anti-HIV positive based on? In a series of preceding reports we have discussed this question in detail, 11 to 14. We came to the conclusion the laboratory finding anti-HIV positive is primarily the expression of an autoimmune activation of the immune system linked to a persistent catabolic state of metabolism. In view of the fact that the diseases grouped under the term AIDS are limited to risk groups such as homosexuals, drug addicts and recipients of blood products contaminated with parenterally transmitted hepatitis inductors, the question is raised whether the anti-HIV test determines autoantibodies directed against cell envelope structures with a specificity to the body's own proteins of the host cells. It has been known for over 20 years that chronically active hepatitis, currently hepatitis B, hepatitis C and autoimmune hepatitis without evident antiviral antibodies, react by the formation of autoimmune antibodies directed against cytoskeletal proteins of the liver cells. Thus the raised anti-actin autoantibodies are pathognomonic for chronically active hepatitis. 15. Johnson et al. In 1965 were the first to report on anti-actin autoantibodies. 16. They described autoantibodies directed against smooth muscle cells and showed that this had to be considered as characteristic indication of leukoid hepatitis. In 1973 Gabbiani et al. Demonstrated that autoantibodies directed against smooth muscle cells react with actin-containing microfilaments. 17. Further investigations indicate that autoantibodies with anti-actin specificity are to be classified within the big group of autoantibodies against filamentous proteins of smooth muscle fibers. 3-18% of healthy individuals present low-titer autoantibodies against cytoskeletal proteins. 18. High-titer anti-actin autoantibodies, on the other hand, are only found in patients suffering from chronically active hepatitis and/or biliary cirrhosis. 19. In 1994 Burmaz et al. Showed that both sera from patients with lupus erythematosus and from mice suffering from the same illness react with glycoprotein 120 and peptides of the postulated HIV-1 envelope. 20. They further proved that control sera of healthy individuals and patients with other autoimmune diseases contain small amounts of the same autoantibodies. Last but not least, they showed that autoantibodies reacting with glycoprotein 120 do not possess antinuclear specificity. They refrain from investigating a specificity against cytoskeletal proteins of these autoantibodies. Evidence is given that the anti-HIV test does not indicate antibody formation against the postulated retroviruses as, during the last decade in Germany, not a single seroconversion has been observed in in-person drug addicts. All seropositive drug addicts acquired their anti-HIV positivity before their imprisonment. In opposition to this, seroconversion by hepatitis B inductors was recorded in intravenous drug addicts, 21 to 23. The same was also observed in hemophiliacs, i.e. despite continuous substitution with hepatitis-contaminated blood products. Approx. A third of these individuals never become anti-HIV positive. This is characteristic for the individual response to autoimmune reactions against cytoskeletal proteins in the host cells, in which Gerard and Sen?Cal observed a polyreactivity. 24. The individual autoimmune reactivity either appears at first contact or fails to appear even at multiple contacts. We conclude that a positive anti-HIV test does not indicate an antibody formation against retroviral HIV antigens. Low-titer anti-HIV antibodies